What is it?
Put it ran through the wall, probably getting people tiny. I've never been literally for a while. No, I've only held like domesticated ones.
It's that time of year, y'all. They'll be in this building off and on.
Yay!
I don't know what the deal is with that, but it's a thing that happens, so.
I should have stopped and got I was so afraid of being late, because when I got on the highway, I like, normally it's not bad. It was like immediate, like stop traffic. I was like, at 9:20. Yeah. I figured it's normally not gonna be okay. Maybe they're having trouble with here. Yeah, I really want to that would be another thing. I thought, well, yeah, they can do it, you know, as yeah, and it goes to like every all the time. And then I did also last week. Yeah, I can do that.
You Dr. Young.
Okay.
I don't know if you ever like mess around with vintage games much, but I got like a vintage game. It's counterfeit.
Counterfeit.
It's counterfeit. I don't really mess with vintage games much. So when I got this, I didn't realize until I compared it to my authentic copy of Soul Silver and realized that this color is different. And it feels weird on the edges and it like comes out of the color slot.
Crazy.
But it works perfect.
Yeah.
I can literally just open it up right now and it works perfect.
That's crazy. I've never seen anything like that, but I mean, I believe they exist, but I've never.
What counterfeits?
I've never caught it.
Basically, they just put the ROM into a normal, like, regular DS cartridge, and then, like, sell it and pretend it's the real thing. See, it works. It works perfect. And I got my team and stuff. This guy. That's about it. Yeah, it's perfect. That's weird. So, someone told me that if it starts splitting out too much, to just trade all my Pokemon to the authentic copy, because this can trade to the authentic copy. The only thing it can't do is it can't interact with the Pokewalker.
Yeah.
Which, well, when was the last time you used a Pokewalker?
You didn't even know. I don't even know what you're talking
I
about.
was maybe like 6. I vaguely remember having a Pokewalker when I was maybe like 6. It was like this weird thing that you could use with your DS on Pokemon Gym board games, I think, or something. I don't know. I don't think it really became that popular. I feel like I had one when I was like maybe 6 or something. That's all I remember. I wasn't intending on using one. There's ways you can tell this counterfeit. I'm just stupid. I don't know.
Okay. So I'm gonna call names like we're, you know, back in the 90s or something, since I don't have a sign-in sheet. So I have Marissa. Okay. I have Cynthia or Cindy. All right, Stephanie.
Here.
Sarah S. She sounds even late. Ariana. Okay. Grace.
Hi.
Corey.
Here.
Okay. Valeria. Kylie. Okay. Melissa. Leanne. She's not in yet. She's not in a little bit later. Yeah. Abriana. Right, Ingrid. Tammy. Lauren. Laquita. Sarah H. Olivia. Kaylin. I think we lost. Renee. Madison.
They were out there.
Angie. Angie's not.
She went late.
Yeah, there's like a few people that kind of scatter in late. There's Leanne. So that's all right. Sorry, you guys are coming in late. I don't have a sign-in sheet, so I just want to be sure I get everything. So we said Eileen's here, right?
Yeah, she's right there. I think she was, yeah.
Okay. So we're just waiting on Sarah S. and anybody, I know you guys, I got you guys. Okay, Elizabeth, right? Okay. This was an old list, so forgive me. Good. Okay, cool. Good enough. Let's get started. So this will be chapter 10 review. Remember, you guys can join in the middle if you'd like. Oh, let me lower this. No. All right, which term refers to all of the chemical reactions that can occur in the cell? So metabolism is just referring to all of it all at once. Remember, there's two different terms that fall underneath metabolism, which are anabolism and catabolism. So what is anabolism? Close the store. The actress is not want this. Like I just saw my student. All right, so anabolism. So between anabolism and catabolism, anabolism is where we're going to be building things, so creating bonds. It requires energy anytime you're building. All right, enzymes are an example of what macromolecule? Exactly, they're mostly, almost all of them, and definitely for all of our purposes, they're proteins. Where does the substrate bind to on an enzyme? All right, so it's called the active site. Good job. What are causes of a denatured protein? So this one's going to be pH, heat, and there can be chemicals that can denature as well. We focused on the pH and the heat the most. When we get into chapter 11, we're going to talk about some chemicals that can do that as well. What best describes non-competitive inhibition? So non-competitive. All right, so in this case, non-competitive just means we are not binding at the active site. So the site that it does bind at is either called the regulatory or the allosteric. site. So both of those are the same thing. Just some terms to be familiar with. All right, the 5-carbon sugar that we find in ATP, what's that? Knowing our structure of our ATP, we know there's a base, we know there's a sugar, we know there's phosphates, of course, but what's the sugar? All right, so this one is going to be ribose in this case. It's just a nucleotide with extra phosphate. That's all. So it's like a ribonucleotide. Think of ribose as like the basic one. And then deoxy is the complicated one. All right, aerobic respiration requires what? Just to be clear, because I don't know why, but this is one of, in case you guys didn't watch the review that I did on Friday. I know it was a long one, but maybe we can go over this review. It was in the, maybe I don't remember. Anyway, anyways, chapter 10. Yeah, it was chapter 10. Did I review chapter 10? Yes, it was. Okay. Not imagining it then. Okay. It's like I thought I reviewed chapter 10. Yes, I did. Okay. One of the most missed questions for some reason is whenever I asked people about what the final electron acceptor is in aerobic respiration. So we're talking about 3 types of ways to make energy in chapter 10. We're talking about aerobic, anaerobic, and fermentation, right? Those are the only 3 that you need to ever worry about at all, ever. So the only thing that they're differing in is the following. We have fermentation that just stops at glycolysis. The only reason it does that is because normally it will go through aerobic, but whenever oxygen is available, it's going to go through fermentation. So it's like a stunted version, okay? So it's the weird one at the end, but anaerobic and aerobic are the same except for their final electron acceptors. And those are differentiated in the name. So if it helps you, if you hear aero, if that's not enough to tell you that it's air, you might be thinking like, oh, but there's nitrogen and a nitrogen, blah, blah, blah. Don't overthink this. Think of it as being like this. Air, the O, think of that as being oxygen. That helps at all. Okay, if you hear aero and it's not got an in front of it, that means, we should know by now anyway, but this means without. So if it's aero, air, O, oxygen, your final electron acceptor, okay? That's what defines it as aerobic versus anaerobic. Literally that is what defines it. Only that. That's the only difference. So if I'm specifying aerobic respiration, you should automatically thinking that's the one that means oxygen. It should be just automatically in your brain. I feel like it's in the name of it, but I don't know if it's the questions that, people get stressed out, right? I think that's really what it is. And then we lose our train of thought. The gas produced by yeast in alcoholic fermentation. So we're fermenting. This is a possible gas product that you can get. It makes our beer fizzy. We talked about this as being a byproduct of how they deal with that pyruvic acid and the electrons that are left over. One of the products that can happen is carbon dioxide. Of course, you can have ethanol, ethyl alcohol, but that's not a gas. And what was it? Well, lactic acid or other acids as well, but those are not gas. Okay, during photosynthesis, plants use what gas to produce glucose as their food? Say as their food, as their carbon source, right? So if they're trying to make glucose into their organic carbon source, what gas do they need to do that? Why this one is so much time. I don't know how that happens. All right. So this is carbon dioxide. They're going to be using carbon dioxide to fix it into our organic molecules. like glucose, and then glucose they're gonna be shuttling into aerobic respiration, going through the mitochondria and all of that, because they have mitochondria too. I have to keep reminding people that plants also have mitochondria. You don't ever just have chloroplasts. So good work, guys. So get out of this. Good job, everybody. We'll start with chapter 13. Okay, so we're going to be talking about disease, basically, the development of disease and how it presents in our bodies. We're not going to not getting into how symptoms are generated. We're not talking about that yet. That's all immunology. If you guys aren't aware, 99% of any symptom that you have in any sort of illness that you're going to have anyways, is going to be from your immune system reacting to it, not from the thing causing problems, but your immune system reacting to the thing. So most of the time. So we'll be getting into that in the next unit. But as far as how we look at disease and, use some of the terms to analyze the different, pathogenicities of these organisms, that's what this chapter is kind of going to get into. So the first thing we need to understand from all of this is that we have a biome other than just our body. There's other creatures living in our body. And those are going to be like our microbiome in our gut or on our skin, in your mouth. There's microbes that are, on your scalp, wherever they could be. These are what we consider a normal human microbiome for the most part. And if they normally are expected to be there, then they're not any pathogen or anything. They're just chilling. Yeah, we're fine with it. They can be commensal, or they can be, I feel like this is, I'm not sharing my screen. That's what the problem is. Sorry. Share. Here we go. There we go. All right. Oh, wow. Sorry about that. All right, there we go. All right, so the holobiont is the term referring to you plus all of your microbiome. That's a term that I'm probably never going to ever bring up again after this chapter. All right, so infection refers to a condition where we have pathogenic organisms that are basically multiplying when they shouldn't be there, okay? So that's an infection. And then disease is different, right? You can have disease without infection. And then you could also technically have infection without disease. It's not very common, but you could. So that would be something growing out of control, but your body hasn't started creating any side effects or anything like that. That's not. We consider disease to be any deviation from health. Right. And yes, I understand that that is subjective, but that's true. It would be subjective because if normally, you're a relatively healthy, active person and you're doing well overall health-wise, then any deviation from that could be considered a disease. But if somebody else their norm is like, well, we're kind of like, sedentary and we have pain and normal, whatever, but deviating from that would be considered disease. So yes, it is a vague, subjective term on purpose. Okay. So disease is deviation from what you would consider to be normal. And you can have that without infection. Diabetes is an example of that, right? That's a disease for sure. Cancer, that's an example of that. You don't need infection for these guys, right? All right, we're going to talk about the normal biota. It can include pretty much any organism in your biome, okay? We know it's bacteria for sure. We're familiar with that. We also should be familiar with the fact that you can have fungi as part of your normal biome. When I say it as it being like yeast, right? So that's okay, then that makes sense. Because we know we do have yeast as part of your biome. It should be there in small numbers and in controlled numbers. And all of these guys are going to operate with one another to help maintain, healthy levels of these guys so that we're not getting out of control. Because you can be commensal, you can be, it could be parasitic, it could cause problems, mutualistic, we're both benefiting, things like that in our symbiotic relationship with these guys. And then, of course, you can also have organisms that could just be passing through. Something, let's say you ate something that had organisms on it and it didn't make you sick. They were there, but like, I don't know. But so these guys are part of our biome: bacteria, fungi, protozoa, viruses. The only thing missing from our list of possibles would be the helminths. that should make sense to people. Yeah, we don't have, worms normally in our body. TikTok might tell you otherwise. That's not true. I think I had this. I told you guys about this, didn't I? Did I tell you about that? So for those of you who don't know about TikTok and how they like to make up lies to, to get views and whatever. There was for a while a thing going around where, people were posting videos of how everybody has worms. And like, if they would do like this diet or whatever, like the lemonade with the cayenne pepper diet, it would help clear the worms out of your body and you could see them. Or you could just take ivermectin that was meant for like animals from over the counter to help clear out the worms that are in your system that the doctors are like not telling you about or whatever. It's this big old like, it was a TikTok generated conspiracy theory. It's probably still around. I don't know what the deal is with it, and that people try eventually, of course, trying to sell their own thing to help you with your worms that you have that your doctors aren't telling you about. I can assure you that with the number of colonoscopies that people are getting, that we would have known about this horrible problem. They give you pictures of your colon. I don't think they're just pulling those off of the internet and giving them to you. Otherwise, I don't know why, if you were a proctologist or if you're like a GI specialist, like why you would be dealing with people's butts if you're just going to make up the answers anyways. Like seriously. So I don't think there's worms in there. I think that would have caught it. I am also, most of the times whenever people would treat themselves and like see worms like in the toilet or whatever, it's the lining of your intestines fluffing out. And that's the truth. People who have Crohn's disease or IBS or ulcerative colitis have probably seen something like that just as part of their disease process and can tell you that their doctors have told them that's the lining of your intestine that you're shedding and that's unhealthy. So these people are actually forcing their bodies to shed that lining and then saying, oh, look, it's a worm, when it's not. It's just your protective cells of your now you're making yourself worse. Anyways, so no worms in your body. It shouldn't be there. Whether it's causing disease or otherwise, there should not be worms. Everything else could be part of your normal biome, okay? Not every organism, but guys from every category. So that's what I'm saying. So humans are colonized by trillions of microbes. They could be resident. So that's part of our biome. We could be potential pathogens. They could cause disease. And then transient microbes that are just kind of passing through, like if you ate something or drank something, or you came into contact with somebody that had staph, but it didn't cause a disease on you. So remember that whole structure is going to be called the holobiont. Anyways, infection means that whatever the organism is actually growing to a point above what it should be. Okay, so even if it is like, let's say you harbor staph naturally, and like you take some antibiotics and get rid of other normal bacteria on your skin, now the staph can overgrow and then lead to presentation of symptoms and the like. There, okay, so thanks. Sorry. All right, so things can overgrow. Right, women know all about this with like the yeast infection, UTI, back and forth BS. So if you have a yeast infection and you're treating your yeast infection, it can lead to bacterial vaginosis because you're killing off all of the yeast that were helping maintain that environment. So now you have BV, or if you're treating UTI, which isn't BV, right? That's just a UTI. You could be killing off the normal biota of your genital area, like the yeast, or sorry, the bacteria that are normally supposed to be there, like the lactobacillus and things like that, that are in very high numbers. You kill those off and the yeast can overgrow and then you get a yeast infection, which is why your doctor will remind you if you get put on antibiotics for a UTI, and they'll tell you, okay, have you ever had any problems with yeast infections when you take antibiotics for UTIs? Because they'll want to look out for that. This is common, and a lot of us are aware of this. Those bacteria and the yeast are typically things that are found in your body normally, even the ones that cause the UTI. 80% plus of all UTIs are going to be caused by E. coli from your own bottom, usually, right? And that's why we say wiping front to back, and I'm being serious about that. So, or like, when you're having sexual intercourse with somebody, there's a lot going on down there that can move stuff around. So then they recommend that you urinate after sex and maybe wash or whatever you feel like is appropriate for you. But to help prevent that, and even still, people get UTIs. So, anyways, this would be bacteria in the case of E. coli that belonged in your bottom and then got into the urinary tract where it didn't belong, right? So it was a normal part of your biome. It is a normal part of your biome, but not in the urinary tract. It shouldn't be there. So now we have something that was part of your biome that was resident turning into a pathogen. Does that make sense? So that usually it's going to happen if we move it into a place that it doesn't belong, like we see with E. coli in a urinary tract infection, or if we have decreased the population of other organisms in its environment around it. You go into the hospital to have your hip replacement, or you go into the hospital to have any other kind of surgery, and they put you on some pretty heavy antibiotics. Naturally, in your colon, you may have something called Clostridioides or Clostridium difficile, right? And that's C. diff. That's a normal part of your biome. A lot of people don't know that. Not everybody has it, but some people have that. It's a normal part of their biome. You kill off all the other bacteria where you only had a couple of guys, a couple of seats in the stadium were taken up by C. diff. The other seats were all taken up by normal things like Klebsiella and Enterococcus and all these other things, and normal E. coli and whatever. But you kill off those guys, C. diff is going to take over all the seats, right? Now you have an infection. It's gone beyond the population, what you should see with C. diff in your colon. And that's when you start seeing disease develop from that. So remember, disease and infection are different things. Infection can lead to disease, but disease doesn't always rely on infection, because that could be like diabetes or something that's totally unrelated to infection. So you can see where all these terms are starting to differ, and we're getting to understand the differences between them. How you can get, of course, we know you can get bacteria from outside of your biome that can make you sick. You don't need me to tell you that. But remember that the ones in your biome, let's not forget those guys, they can move to different places or grow out of control, the numbers that they should be, and that can lead to disease too. Yes.
Did you say that you're not supposed to urinate after sex?
You are supposed to.
Oh, okay.
Yes.
I was gonna say, like,
To flush it out, yes.
To
To flush out any bacteria that could have gotten into the opening of the urethra, you're supposed to, yes. And if you guys haven't been told that, that's a thing. So just, you learned a thing, I guess, today about taking care of your bodies. Anyways, don't we all wish that following all these rules worked perfectly 100% of the time? Believe me, I wish that they did too. We have the Human Microbiome Project looking into all the possible microbiota that you can have in your body. I'm not going to go over the details of these, okay? But you definitely want to have a balance of what types of microbes you are supposed to have. No one guy should be out of proportion from what is expected, because that can lead to potential infection and disease. Anyways, there's things that can make you more likely to get infection. I feel like these are obvious, and I'm not going to go over them too much, but age, genetics, pregnancy, surgery, anything that can weaken you at all, even if it's genetically related, can increase your likelihood of infections. Endogenous infections, this is a term referring to when you get an illness from bacteria on your own body. So this is talking about E. coli getting into your urethra. That would be an example of this. Another one they're getting here is Streptococcus pneumoniae, which can be part of your normal biota. It'll live in like your nasal cavities and things like that. And then it can cause pneumococcal pneumonia. It says in AIDS patients, you don't have to have AIDS to get pneumonia from this guy. It's the #1 community associated cause of pneumonia, and the #1 community associated cause of meningitis across the world. So this is a biggie. Streptococcus pneumoniae. If it's present in like your, upper respiratory tract normal numbers, you're fine. If something happens to shift that, or maybe you didn't have it originally, or it became resistant to an antibiotic, any of these things can cause it to lead to disease, like pneumonia, like meningitis. A lot of this can also be usually in tandem with having some sort of weakened immune system situation. You got sick with something else and you get a secondary infection. All right, where do babies get their microbiome? So when you are growing up as a baby in a womb, you're just cells that are populating. The bacteria that are in your mom aren't necessarily populating along with your growing cells. So at which point are you really going to get exposed to the bacteria that are going to become part of your body as you grow? Well, we do think that the first place that you're going to, actually they've shown that the first place you're going to get exposed is going to be in the uterus. It's just not going to be growing along with you as you grow, right? You're going to be kept in your little happy little placental, area, and have your own blood flow and everything like that. And the bacteria around you should be maintained and kept, kept from growing out of control is what I'm trying to say. by the mother's, womb and her immune system and everything like that. However, you will get exposed to it, and that's usually where you're going to get your first introduction to microbes. It can be part of your biome. Definitely the most recognized point at which you're going to be exposed to even more microbes that'll be part of your biome as a baby will be through birth. And of course, if you are being born through vaginal birth or through C-section, you're going to have very different experiences with what bacteria you're going to be exposed to, right? We know that if you're being born through C-section, they're probably going to keep the area as clean as possible. They're going to disinfect it as much as possible. Try to make it sterile. It's not going to be 100% sterile, but that you might see some bacteria from that. But there's going to be a drastic, difference from what you're going to see going through the birth canal. There's going to be a lot of bacteria. In fact, there's such an issue with the bacteria in the birth canal. We'll talk about some of the sexually transmitted diseases in Unit 4, that you can be exposed to bad bacteria that were in the birth canal. The babies can become blind or get, lifetime infections from just having passed through the birth canal and been exposed to the microbes there. The mother, if they're carrying an STI or something. Okay, so then we have the milk, of course. If you're drinking breast milk, then there's the biome from the mother that can be passed on to the baby. The breast milk has bacteria in it that will help the baby. They'll colonize the baby's gut and help it be able to metabolize milk. It can't digest the milk otherwise. So it needs those bacteria. And if your baby is on formula from the get-go, that doesn't mean bad things. It just means you have to have a formula that has probiotics in it. So bacteria that are going to be healthy for the baby's gut added to it. And a lot of them do, so that you can get a healthy kind of gut biome started there. If you don't do that, then you can see that your baby's going to have a lot more GI problems growing up. So then, of course, the more you're exposed to other people, you're going to get to see their bacteria and incorporate that. And then your baby can also pick up the bacteria from their environment. Now, are you done picking up your biome once you are a little baby and you've got, it's like you fill up all the stadium seats and that's that, and you don't need any more different bacteria? And no, obviously, right? So let's say that baby goes through puberty. That baby's changing, and let alone the things that it went through to get to that point. But you go through puberty, that hormonal change is going to cause a shift in your microbiome, just because of all the different levels of pH changes and all the different hormones and all the things going on in your body that are different and all that. Then, of course, if you're going to be having sex, once you start having sex, you're introducing like your entire reproductive tract to somebody else's reproductive tract, that's going to introduce new bacteria in that area as well. And it will be shifting pretty much for all sorts of reasons, even beyond that, throughout your lifetime. And as you get older and your immune system gets weaker and everything like that. So that's the idea of your microbiome. It's important for you because it takes up your normal microbiome, your healthy microbiome, the way that it's supposed to be, should be taking up all the stadium seats so that bad guys can't sit down. That's the idea. Okay. That has its own term. It's called microbial antagonism. I think that it's in this chapter, but I just haven't gotten to it yet. I'm gonna write it anyways. No, I'm not. Microbial antagonism. I just want to kind of get this one going so that we can get through both chapters today. But anyways, a pathogen is where there's a parasitic relationship with the host and the microbe. True pathogens always cause disease. So they can cause disease in healthy people. So if it, like the plague, if I brought the plague in here in some fleas, right, and let the fleas bite everybody in here, it wouldn't matter how healthy you are going to be susceptible to getting the plague, right? If I bring Ebola in here and just wipe you all down with some Ebola blood, and you're going to get Ebola, right? However, if we're talking about the opportunistic pathogen, like if I were talking about Candida auris, it can be part of your normal biome. Most people aren't going to see it in very high numbers at all, but Candida auris is a huge problem in people who have weakened immune systems, especially if they're on Humira or something like that. They can have severe yeast infections all over their body and infiltrate their skin, everything like that can get out of control because of that weakened immune system. So these things can happen from our host defenses being compromised and our immune system being weak and things like that. So I also want to be clear. When we say your host defenses are compromised, we're not only talking about your immune system being weak. You could be like you stepped on a nail, and that's what let the endospores into your foot so you can get tetanus. Okay, so that could be penetrating the defenses too. And then things that are normally part of your body that is not natural to them. But yeah, if you have some bacteria that are supposed to be nice and healthy on the outside of your body that get deeper into your body or that start infecting deeper tissues of your body, especially when you have already an inflammatory response going on, it can cause severe damage, such as, just to be clear, if you guys don't know what necrotizing fasciitis. Necrotizing fasciitis is the flesh-eating disease. It's extremely common, and especially people who have diabetes. The bacteria will just start eating away at the tissue of these individuals. It's healthy tissue. It's just it keeps on, infecting and eating and causing it that flesh to die as it goes along. So it's necrotizing. That's just caused by like staph and strep, not a weird bacteria. So think about that. I'm telling you that horrible flesh-eating disease that can like cause people to lose limbs and things like that, that these are caused by bacteria that are normally found on your body or that just cause relatively mild infections. Maybe, get a boil here or there, maybe get a sore throat. That's what's causing that. So it just depends on maybe how deep it gets into the tissue, maybe how it was introduced, what your immune system is doing, and things like that at the time. All of these things play a part. So virulence is a term that you guys always hear me talking about virulence factors. This is not going away anytime soon. Virulence factors, virulence is talking about the degree of pathogenicity. Pathogenicity is a term referring to that it causes disease symptoms. Okay? How pathogenic, how disease it is, how bad is that disease, that's virulence. It's just a degree of it. So this is going to be determined by the ability of that microbe to grow in the host and cause damage in the host. And that can be different from person to person. A virulence factor is anything in a microbe that helps it establish pathogenicity, so cause disease. If it is something that helps that organism colonize and cause disease in a person, that's a virulence factor. It doesn't have to be nasty, crazy things. Like we always want to say, virulence factor, oh, that must be something horrible. Flagella are virulence factors because it helps the microbes in some areas to move around and get to new areas and colonize them. That can be a virulence factor. Obviously, we know antibiotic resistance and things like that, but it could be just simple things. A capsule is a virulence factor. It helps, helps you run away from the immune system. I'm not really going to ask too much about the infectious dose, but this is just referring to the minimum number of microbes for infection to start. Some diseases only need like one. A lot of viral infections only need like one virus to cause a true disease. You just need to get one virus into your body and to get the disease. Low end of bacteria would be like 10. Tuberculosis is a 10. So it's not super, super low, but it's pretty low for bacteria. Some of them you'll need to have like 50. I think salmonella is like 50 or something like that. So 50 cells into your body in order for you to be able to cause disease for most people. So here are the steps involved in what causes disease in a host. You need to get in. That's finding a portal of entry. I'm not going to go over that one, I don't feel like, because, yeah, I don't know. You got to get in. Attaching and negotiating the biome. So what that means is finding a way to attach to whatever surface you're going to grow on, and then dealing with whatever the normal microbes are in that area, the stadium seeds that are already full around you, right? Surviving the host defenses. That's going to be dealing with our immune system for the most part once you get in, causing damage. So whatever you are doing as you are growing up as a pathogen, that you're wreaking havoc around whoever it is that you're infecting, killing cells or calling an immune response with killing nearby cells, all sorts of stuff like that. These things can lead to symptoms which we perceive as disease then, right? Disease is deviation from health. How do you know you're deviating from health if you aren't having signs and symptoms? Signs and symptoms are what define that. And yes, we're about to talk about signs and symptoms, and they are different. You didn't know that. Okay, a sign is not a symptom. Symptom is not a sign. All right, and then you need to get out of the host to go to the next guy. So like I said, our portal of entry, you need to get in somehow. Remember this part at the bottom. Remember, you can come from outside the body or already be part of the body, but move somewhere else or grow out of proportion, okay? If you're exogenous, you came from the outside of the body. If you're endogenous, you were already in endo, the body, and you're just growing where you shouldn't be or out of control. I'm not really going to talk about the baby, the placenta, because I just feel like, of course, you know what a placenta is. It's where the mother and the baby are going to exchange waste as well as nutrients. And this can be affected by bacteria as well. So that can keep the baby safe as well as create a portal for some bacteria to get in. Just depends on the infection we're talking about. So I feel like it's not actually that useful to talk about because you can get infected or not. I don't know. So adhesion, this is where we're going to have our microbes stick onto the surface so we can start dividing, or stick onto, you know, if you're a virus, latch onto whatever your, lock is with your little spike key, and that's going to be your attachment. So quorum sensing, we've talked about this with the microbiome, so they can, talk to one another about what they're doing by creating signals from chemicals they release as they are multiplying. But yeah, here's some mechanisms they can attach by. Fimbriae are appendages we talked about previously in bacteria that allow for attachment. That's what they do. Fimbriae do that. Remember, flagella are for moving. Fimbriae are for attaching. The pili you can attach, but it's mostly used for channels to move DNA and stuff between. Capsules sticking onto things, and then we have our spikes attaching to their target proteins. All right, so we're surviving the host defenses now. The phagocytes are what are going to come along and eat us. We've said them a million times. I don't feel like I need to go over that too much, but anti-phagocytic factors are things that some of these microbes can have that help them avoid phagocytosis. What's an example of a anti-phagocytic factor? Capsule. It's that easy, okay? So anything that prevents phagocytosis, anti-phagocytic factors. So it can be hard for them to engulf. That's what the capsule does. And it makes it so, or you could have some guys like tuberculosis that are adapted to survive inside of the phagocyte. So tuberculosis infects alveolar macrophages. So macrophages are some of our phagocytes that do big leg work for our immune system. So your lungs will send alveolar, like the alveoli in your lungs, alveolar macrophages to the site where you have TB, and then they will eat up the TB guys, but that's where they want to be. They want to be in those. And so they just live there and are happy and say, thanks for letting me in. And that's where they multiply. Their favorite areas. They can adapt to phagocytosis by, hey, this is working out for me. Then will cause disease. These are our virulence factors. So anything that's going to be allowing for pathogenicity, any signs and symptoms, okay? These can be enzymes and toxins we're secreting, could break down things that are holding the cells nearby, like the collagenase that we mentioned previously. So breaking down that extracellular matrix so we can move into deeper tissues, or breaking, or like even breaking into the cells, breaking apart the cells, causing overreaction by the body's defenses that can lead to inflammation and all sorts of things. Altering the host cell genome or transcription process. That could be like when we insert our viral DNA into the genome. That's an example. Taking over that stuff. All right, exoenzymes are enzymes that bacteria will secrete that do work for them. That would include the collagenase that we were saying, or hyaluronic acid, hyaluronidase down there at the bottom. Those guys, collagen and hyaluronic acid, are part of your extracellular matrix that help hold your tissues together and your cells together to make up your tissues. So we can, as bacteria can secrete enzymes that break those down so they can get between cells, cause inflammation in the tissue and things like that. It breaks down your barriers. A toxin is anything that's going to have we say has a poisonous effect, but has any damaging effect to the body at all. That's secreted by usually a microbe. Okay, so exotoxins are secreted outside. Endotoxin is only one thing. Remember, exotoxins, anything. Gosh, why did that do that? Okay, whatever. I didn't click a button. Exotoxin, anything that they're secreting outside. Endotoxin is not secreted, right? It is only ever our good old friend LPS, lipopolysaccharide. When I said lipopolysaccharide is LPS, aka endotoxin, that's the only endotoxin you need to worry about. There's no other. We'll never be talking about any other. Great. So this is showing hemolysis that is accomplished by some bacteria that are growing on blood agar. Hemolysis, hemo being blood, lysis breaking open the blood cells. You can see here that in our enterococcus, this guy is gamma hemolysis because we didn't have any change. Alpha, because we have a greening. Beta is where you have true clearing. And this is from the actual true full breakdown of those blood cells. So we can, this is going on in your body when you're sick with these guys. When we're looking at the bottom here, it's strep pyogenes. This is streptococcus pyogenes that causes strep throat. It'll be involved with rheumatic fever. It'll be involved with, what was I just saying earlier? Necrotizing fasciitis. It can be involved with impetigo. It can be involved with a whole lot of stuff. Like strep pyogenes is a big bad. Having said that, it can be not that big of a deal also. It just depends on your body and how you're exposed to it, right? But they have a hemolytic enzyme to break down red blood cells, and they can do this in your body. So that's why that's an important thing to be aware of. Excessive host response. Here we're just triggering the immune response at a level higher than what would actually be beneficial. And then that can become harmful because a lot of what the immune response does, especially the inflammatory response, that can cause damage to our tissues too. So it's not just causing us to kill bad guys and single them out, but all of the signal that we're sending out to kill bad guys, it's not perfect response. We're also causing inflammation in our own cells, killing our own cells. Inflammation causes damage and all that. So we can't have inflammation for extended periods of time because it can lead to permanent damage. If you guys don't believe me, then look at rheumatoid arthritis, because that causes permanent damage to people's joints. That's just inflammation that did that. So their own bodies causing inflammation in their own joints. You don't need bacteria to cause the damage. You can do it to yourself. You don't need any help. So yeah, a big one too, we have long COVID. Long COVID, people who are having COVID symptoms for a long period of time after they had, gotten rid of COVID, and some of them really weird symptoms too. But that's usually due to an overstimulated immune response. At least that's what we think it is. There's different patterns of infection you can have. You get a little boil or a pimple or something like that. A pimple is an infection. A boil, same thing. It's just not usually where you would expect it to be. And these all have to deal with, there's different terms for those. We'll talk about those in chapter 19, what the differences are in those, but you get the idea. It's localized. You scrape your knee and your knee gets infected. At first, that's localized. Should stay that way, hopefully. Systemic means it's going to be passing all over in your body. And here we're talking about things like sepsis. Okay. Focal infection, if you got a boil and the boil, you popped it or whatever, and then you spread it to other areas of your body. Now it's not just in that one area, now it could be other areas because you're not keeping it clean or whatever. That's an example. Or in this picture, like it's showing, it has the nose to the heart. If you have strep infection, that can pass on and cause rheumatic fever, which is dealing with your heart. Like bacteria can infect your heart, stuff like that. Move to other parts of your body. Then we have, so not everywhere, but just like from one place to a new place. Everywhere is systemic. Mixed infection means there's more than one microbe involved in that area. And you could have a primary infection, like you had a UTI, and then you get a secondary infection as a result of having that first infection or treating it or anything like that. All right, so I'm not going to go over this. We just talked about it. All right, acute versus chronic. You guys know what that is. Short, it came on fast, it's acute. Long time, chronic. So let's talk about signs and symptoms. Signs, any objective evidence of a disease noted by the observer. Symptom, these are subjective. So I could say these are objective and these are subjective. And if those are vague words for you, and I would understand if they were, I get it. So signs are measurable. Okay. Symptoms are reported. So an example would be a fever. You can measure a fever, yeah. If somebody has swollen lymph nodes, you can feel their swollen lymph nodes. If they are actually vomiting, you can see that they're vomiting. If you can actually verify that the thing is happening somehow, that is a sign. If it is only reported, that's a symptom. So symptoms would be things like pain or nausea. You can't verify that. The vomiting you can, not the nausea though. Fatigue. Those are biggies, right? Pain and fatigue. Those are probably the two biggest ones that somebody could come into the ER and be saying that they have pain or that they've been experiencing severe fatigue. And you're just like, Are you sure, or are you lying to me? And you can't prove it, right? How would you? If it was that easy to prove, then believe me, ERs all over the place would be thanking you every day for having proved it, because these doctors have to treat people as though they actually have real pain, even though they're coming in and looking for drugs. They just want, morphine or whatever, and you have to treat it. So, whatever. And then you can send them out with a pamphlet to, get your drug addiction, go fix that. But like, that's the best you can do. You can't turn it down. So anyways, so signs and symptoms, measurable versus reported, right? So of these two terms, if I were to say, if I had a sign in my hand, like an actual sign, like a stop sign or something like that, that's a physical thing that I can touch and is tangible. A symptom is a word that I can't have. Can I take a symptom and no, right? So that's like how I think of the difference. It's like sign. Okay, I can actually have a sign. Now I know it's a different thing, but you could physically, that's a thing you can interact with. That can measure that. That's how I remember which one's which. All right, this bottom term down here has nothing to do with either of these, but it's on here because it's an S, I guess. I don't know, but here it is. Syndrome. These are diseases identified or defined by a certain complex of signs and symptoms. So what I mean by that, we've already been talking about one this whole time, a lot of the time. I will bring it up. If I have light sensitivity, if I have a sore neck, if I have a fever, if I have fatigue, these things are signs of meningitis, right? And it should automatically be, and if you guys are going into nursing, and you're going to be doing rounds in an ER, you're going to need to know that immediately, like these are the signs of meningitis, because that's a big deal. It's one of the things you'll learn about there. We do rounds there, but anyways, meningitis. But if I'm talking about COVID, COVID's caused by the COVID virus. If I'm talking about the flu is caused by the flu virus. If I'm talking about the plague, we're talking about bacteria, Yersinia pestis. That's the only one that causes that, right? Meningitis is caused by several kinds of bacteria, some kinds of fungus. There's viruses that can be involved in that, but they all have that same group of symptoms. So that's a syndrome. Like meningitis is a syndrome. Another very obvious one that we don't think about is a cold. If you have a cold, you go to the doctor and they're like, oh, you just have a cold. It's just a cold virus. A cold virus is not a thing. There's 200 different viruses that we know of that can cause a common cold. So that's a syndrome. It's just the same signs and symptoms caused by a variety of different things. Make sense? So this would be, we'll just say on here, just so for sake of, I don't know, future people watching this, meningitis, or it could be a cold, all these things would fall into this category. And yes, there are non-infectious examples of syndromes we could go into that can be genetically associated and stuff, but we're talking about stuff, microbiology here, okay? So we already kind of went over this, but signs are measurable. You can listen to chest sounds. Leukocytosis and leukopenia, whether or not you have too many or too few white blood cells, you can measure that. You can see that there are abscesses, all these things. Whereas the symptoms, chills, I mean, maybe you can see that there's shivering going on, but you can't tell if it's chills, right? And pain is a big one, malaise, fatigue, having chest tightness, you can't measure it. All of these things that are unmeasurable. Headache, man. All right, so inflammation, when you have inflammation starting in response to a disease, the first things that you're going to see are edema, which is accumulation of fluid. That's usually the cells being brought into the area. They come with fluid. Granulomas and abscesses are areas where we're walling off infection. And then lymphadenitis that's lymphadenitis. Anytime you guys ever hear itis, this is inflammation. Always, that's what that means. Actual inflammation of the that's like the adenoids, okay, the lymphatic tissue, okay? So these are swollen and inflamed because they've been activated to respond to infection. Okay, so you can feel that. That's swollen lymph nodes. These are other things we can look at. Leukocytosis is an increase. Leukopenia is a decrease. Septicemia is where you have microbes growing in the blood. Bacteremia is usually only small numbers of bacteria. So if you were to realize that somebody had a little bit of bacteria hanging out in the blood, you should have nothing in your bloodstream. It should be sterile. So if there's any bacteria, that's bad. If it's growing and flourishing, that's septicemia. So it's septic. That's bad. So those are different, so remember that. But, and then viremia is where there's viruses. And then of course, fungemia would be fungi. All right, you can have asymptomatic, subclinical, or inapparent infections whenever you don't have obvious signs or symptoms. That could be due to, not having as much growth. It could be confined to a certain area, like we had talked about with typhoid Mary. Typhoid Mary walking around, spreading around her typhoid. She had an infection in her gallbladder, but she didn't have symptoms of having typhoid fever, which is salmonella. It's a bad form of salmonella, deadly. She didn't have symptoms because it was only confined to her gallbladder, but it was getting out enough that she was able, when she wasn't washing her hands, to spread it to other people when she was serving them food. And so typhoid Mary was spreading typhoid to people that way. I don't know what to tell you, man. She didn't want to wash her hands. She was just against that. And then once you've infected somebody, now you need to get out so you can move to the next guy. There's a lot of different ways you can get out: urine, feces, coughing, sneezing, insect bites, removing blood, manually maybe, sharing needles, for example, stuff like that. That reminds me, I can't wait till we get to that part of the course to tell you guys about needles. But all right, so we've already talked about latency with some of our viruses. Latency is just whenever you go dormant with an infectious agent. It could be actually hiding out in your DNA, but it doesn't always have to be. It could just be at a low level in the background, just like a couple of cells, but they're just like almost hibernating, if you will. That happens a lot with tuberculosis and syphilis and stuff, and malaria. So they can go into latent stages. They're not incorporating their DNA into your DNA. They're just cells like not multiplying actively. So very common in tuberculosis to have that for years. So latency is just a dormant. You don't have any symptoms or signs. Sequelae is a term for long-term damage caused by having an infection. So if you have meningitis, you could become deaf as a result of the inflammation associated with it. Strep throat can lead to rheumatic heart disease, which we already talked about, as the body is responding to the actual bacteria in your throat infection. It can cause antibodies that match your own tissues, and that can cause heart damage, literally. Or you could even have, I guess, movement into the heart itself with the infection. But anyways, Lyme disease can lead to arthritis, and polio can produce paralysis. I feel like polio is the best example of obvious sequelae that we don't have it anymore in America. But as far as obvious, right? So even FDR had effects from polio. In his wheelchair, wheeling around, he didn't have active polio. He'd cleared it already at that point. But he had paralysis forever, permanently, because he had polio earlier on. So if you get polio and you become permanently paralyzed from having had it, and you don't have polio anymore at that point, but you're still paralyzed, that's sequelae. That's not like, okay, that's just how you are now, and that's just how we're going to treat that as. That's sequelae because it's long-term damage from having had the infection. That's the term for it. Okay. There's all sorts of stuff that have stuff like that. We'll talk about polio later on, but polio is kind of fascinating, I think. All right, the course of an infection, that's going to be the incubation period. And interestingly enough, this aligns pretty well with our bacterial growth curve that we introduced in, what was it, chapter 9? Yeah, it was the end of chapter 9, right? Incubation period is the time from contact with the infectious agent to the appearance of the first symptoms. The incubation period is the time where you get the COVID inhaled into your body, and it has to attach and find where it's going to grow and start multiplying and everything, and before you start having symptoms. So that can be a couple of weeks, So that would be the incubation period. The prodrome, or the prodromal period, which a lot of people will hear them calling it the prodrome. These are when you see the very first symptoms. So it's like whenever you get a cold or something and you start getting that tickle in your throat and your throat starts to hurt just a little bit, that's the prodrome. Okay. Acute phase is where it's just all full-blown illness. So that would be you got your, a little bit of a fever, or maybe not, depending on what kind of illness you have. You're feeling like crap, you got the malaise, you got the fatigue, runny nose, all that. Now that's during your acute phase, greatest virulence, right? And then eventually you will go through convalescence. And convalescence means you're recovering. So here's a little bar graph of that. Incubation period, where you don't have anything happening. Prodrome, the first symptoms. height of infection. We'll have the acute phase. This will sort of plateau once you've reached, you'll just have terrible symptoms for a long while, but they're not continuing to get worse the whole time. And then we will have convalescence. And remember how this would associate possibly with the lag phase. This whole part might be the log phase or the exponential. And then this would be the stationary for the growth curve. And then this would be the death, right? We can see how that could correlate with microbial growth. So that's bacterial I'm talking about, but a lot of microbes follow that same trend. There's a reason for that, right? And it makes sense if this is how you're dealing with the microbe and whether they are super grown up or they're not growing as much or they're dying. Of course, that's going to associate with your symptoms. Sometimes your immune system is what's getting rid of them and they're not just dying off from nutrient loss, but either way. A reservoir is a term for where a pathogen would be coming from originally and it just stays there naturally. It could be human or animal, soil, water, plants, wherever it comes from naturally. The source is where an infection is acquired from. That's a different thing. So if the reservoir is in, let's say Ebola exists in bats, right? They say it's probably bats in the Congo jungle or something. And monkeys in that area come into contact with these dead bats and maybe eat them or whatever it is, and they get Ebola. And then the tribes out there go to collect bush meat for their tribe so they can eat, and they will kill monkeys, the slowest ones or the easiest ones to kill. Okay, maybe they're just slow because they have Ebola. And so that can happen. And so that would be the source for that tribe, would be that monkey that they had brought in as food source that could have spread Ebola to that tribe. It's usually how we see outbreaks, starting with that. So that's a source, but is that the reservoir, that one monkey? No, the bat population was the reservoir. All right, a carrier is anybody who shelters a pathogen and can spread it to others without knowing. And this is a great chart that summarizes different ways that can happen. People who are asymptomatic, that have the infection but just aren't showing anything, like herpes when you're not having any lesions. Okay, you can still transfer herpes when you don't have an outbreak. It's just not as likely, but you can still. You're asymptomatic carrier. Incubating carriers, you just got infected, you are infectious, but you don't have signs yourself. Take a long time for some of these things like COVID to appear. It can take 14 days to have symptoms of COVID, but in the first 5 days, you can be contagious. Convalescent carriers, people who are recovering and think they're doing better. How many times are you guys going to go out into public and you thought you had a cold that you are getting over? And you're going to go out in public now thinking that you're all better. You might have a little tickle in your throat still. That's convalescent carrier. You're getting people sick with that. Does that mean that you have to stay home? I mean, you make your own choices, you're adults. All right, chronic carriers, people who have had a long-term infection, especially with tuberculosis, where you don't have a lot, like you have latent infection, latent TB. And then passive carriers, these are people like I come and handle this patient, and then I go to the next patient without changing gloves or washing hands or something like that. All right, we can also have, all right, I guess it's talking about the living reservoirs. Like I mentioned bats or whatever, but this can be animals other than humans or arthropods that could be transmitting it to humans or via vectors, like the rats that had the plague. Their vector was the flea to the humans, but they were rat reservoirs. All right, good plague. Who knows? It's so itchy. The allergies. I don't know what the deal is this time of year, but it's the worst allergies I think I've had in a long time. All right, I don't know. And then people who are actively ill, you can have indirect and direct. Okay, so direct, I sneeze and you guys can say, you got it from me. Indirect, I'm up here with my pen. I have a tendency to put things in my mouth and stuff whenever I'm talking. If I had this in my mouth and somebody else were to come in here and use this and touch it and now they got COVID or whatever, right? So that is indirect. That would make this pen something called a fomite. That's one of the terms you guys will have to know. I think we're going to bring it up later, but. All right, human carriers can be reservoirs if they're infected and maintaining a population. And arthropods are probably very common, most common ones we can think of. An arthropod is a biting insect. So if it bites and drinks blood and it can transmit a disease to you is what this is talking about. So ticks transmitting diseases like Rocky Mountain spotted fever, mosquitoes transmitting diseases like 1,000 million different kinds of diseases. They have hella diseases, y'all. Really do. And there's biting flies like the tsetse flies. And then ticks, oh, we said ticks, fleas, whatever. All right, let's see. Biological vectors versus mechanical. Biological vectors are actively infected. So if you have malaria, the actual protozoan involved in malaria actually infects the gut of the mosquito. The mosquito is truly infected by it. It's not just carrying some blood that had it in it. It is actually infected with it. So that would be a biological vector. Chickens, like if you go into someone who has had chickens and they have avian flu and you get the avian flu from them, they were sick. They are a biological vector. We already talked about the bats. Bats carry a lot of diseases, you guys. I think they're cute. I do love bats. They have a lot of diseases. All right, mechanical vectors are vectors that are not infected. And what that means is cockroaches, flies, things like that come and run across feces or whatever, and then come and run across your food, and then you eat it, you can get sick. That's what that means. They're like literally physically carrying the things around. Yeah, it's gross. Zoonoses are diseases that are associated with animals. Very obvious one that you guys know that I like to talk about would be rabies. Right, so it's maintained in the animal population that people can occasionally become sick from. But there's a whole list of common zoonotic infections here, ones that you might not even think of, like influenza. Usually comes from birds, I guess chickens, yeah, and swine. I'm not really going to talk about the non-living reservoirs, but some things can live in the environment outside of other organisms. You can get sick from them still. Communicable diseases are things that can be transmitted from person to person, able to be communicated, okay? Communicable. Contagious is highly communicable. That's all that is. It's just a bad version of communicable, okay? So it's contagious, highly communicable. Non-communicable does not arise through transmission of infectious agent from host to host. As far as infectious agents go that are non-communicable, that could be something like I always struggle with this one, and then later on in the day I'll think of one that's really obvious. But if you do fall and scrape your knee and it gets infected, is that going to be? It's not really going to be communicable to person, unless you're like coming and licking my wound or something. Like just typically not communicable, right? Alrighty. Horizontal transmission. If you guys get, just like with the bacteria, okay? Horizontal means between like the people who are already here. So you guys could get sick from me, or we have sexual transmission. We can use fomites to do it. We can do direct transmission in all sorts of different ways. And we can even use vectors between person to person, like with what we see with malaria. Okay, that's horizontal transmission. Vertical transmission is when you get it from your mother, either literally from being pregnant or going through the birth canal or through the breast milk. We already talked about fomites. Fomites are inanimate objects that can, they themselves are not a source of infection. This has not got any infection on it, but I can lick it and then give it the ability to transmit whatever. So that's a fomite, and that makes this a vehicle. So a fomite is a type of vehicle, a means to spread. Oh man, I was hoping I could get through the other one, but I don't think we're going to be able to. Water, soil, air, I'm not really going to talk about it. Droplet nuclei and aerosols just means like when you're sneezing or coughing, spreading it to the other people nearby. Then Robert Koch, we've introduced him before, but he developed these steps to identify organisms that were causative agents of disease. I'm not going to go through the steps. You don't need to actually know the steps, but you do need to know that he has steps to help identify the cause of diseases. He actually identified the cause of anthrax via these steps. Okay, that's it for this one, I think. Yeah, okay. We'll try to get into this one a little, and I might have to bring it up on epidemiology. So for chapter 14, epidemiology is just dealing with the who, what, where, when, and why of infection. Okay, so you do need to know about Florence Nightingale and John Snow. Florence Nightingale, she basically, during the Crimean War, saw everybody was getting sick and dying, and it was because people weren't washing their hands and crap like that. So she instituted rules in the hospital for being more sanitary, and she helped a lot of people survive. So going from it said 32 out of every 100 soldiers were dying from dysentery from being exposed in the hospital, not from the field, not from wounds. So she improved that a lot. John Snow, he saw there was cholera going on in London. He made a map of where all the ill people were. He found that there was a water pump in the middle, and then that water pump, he took off the handle, and then people couldn't use it anymore because he told them to stop and they didn't want to listen. So he took the handle off so they couldn't use that water pump anymore. And so then cholera stopped spreading magically, go figure, right? But yeah, he mapped it out. And so he's considered one of the fathers of epidemiology because of that. And this is a little map and a little picture of the death, basically, pumping water for the little kids and get everybody cholera. Cholera is no joke. It's like having diarrhea to the point that you don't have poop in you anymore and you're just pooping out your water and electrolytes from your body. It's rice water stool is what they call it. And it literally looks like rice water, not brown anymore. Rice water is white, in case you didn't know. If you want to poop that out, people die from dehydration. Severe dehydration. I told you guys about this, about what they do if you have cholera. There's these beds called cholera beds. And it's got a hole in it, and they lay you down on the cholera bed and put a bucket under you. That's what they do. That's the treatment for cholera. Because usually it's already progressed that far that it's not going to help to give you antibiotics. Like it can be a self-limiting infection if you can keep people alive. So they'll give you like an IV. Huh?
Does it last long?
Depends. It depends on like the care and support that you get. You can easily die from it. I think it's like 20% mortality rate or something. Anyways, but usually in like third world countries is what I want to say. It's not usually a big deal here, but all right, these terms, know the difference, okay? Prevalence, the total number of existing cases with respect to the entire population. There's no mention of time. So we're not taking a slice of time here. This is the number of existing cases. Okay, sure, we're looking at that as far as the prevalence, looking at the total number of existing cases, but the incidence, this is a different deal. The incidence is like measuring the number of new cases over time. So how many cases in a month you saw? How many cases this week you saw? That's the incidence. The prevalence is going to be all of that in that entire outbreak that you're looking at, all of the cases that we have. So they are different things. Incidence is like looking at like progression of how many we had this week versus this week versus this week, okay? This incidence is also known as morbidity. You have to know that these are the same thing. Morbidity, so you call something is morbid, okay, but it's not the same as mortality. Mortality is deaths. You're immortal, you're gonna die, right? That's how you remember. People, I don't know, get confused between morbidity and mortality because they are both kind of negative terms, but they're in, like, just think about what the words really mean. Okay, point source epidemics. So these are other terms. I'm trying to go through the more important terms for these, even though point source or epidemics. The infectious agent came from a single source, okay, and from a single exposure. So one exposure, one time. I come in here from fall break and I just sneeze on all of you guys while I'm giving my talk, and then I leave. All of you come back and have COVID as a result. So that's, you got exposed in that one incident. So it's just one time you were exposed, and that one exposure, that's it. So you can all pin it down to one point. That's why it's a point source, okay? The reason I'm going over that is this common source epidemic. So it's giving an example of the American Legion Convention. I'm going to go all the way back to John Snow's story. Okay, those people weren't drinking from that pump in that one day or that 1 minute that he discovered it. Right, this was going on for weeks. These people were getting cholera and getting cholera and getting cholera, but they were all getting it from that one pump. So now we're not one exposure, one time. Okay, single source, like the pump, over a period of time. Weeks or months. We know that they were all tied to that pump, but they wasn't over this one exposure. Like if I sneezed on all of you at one time, right? If I just kept sneezing for weeks and you guys just kept coming in here and people just kept getting sick in and out and whatever, that would be common source. But common source, you'll have one thing they're getting sick from over a varied period of time. Propagated epidemic. This is whenever an agent is communicable and maintained in a population. I like this one because everybody knows flu. When flu comes around, it stays around for a while, and it's maintained because it's getting spread within the population. People are just maintaining it. This is why we had to have social distancing with COVID, is to stop that, right? We didn't want to keep people maintaining it in the population, so we had to wear masks. We had social distancing. We had like, don't go out if you do not need to, and all that to stop this from happening. And now we have this with COVID, but we're vaccinated, so we can survive it a lot better. We're not as worried. But yeah, you guys get this one. All right, the index case, that's a term. This is mostly terms, FYI. The index case is the first patient that ever had it. An endemic means that something is, so endemic, think of endo. Endo means in, right? So it's something that is normal in that population. I would think of like a cold. We're always going to have colds, right? At certain levels, right? If everybody started showing up with colds all the time, that would be concerning. But people having colds here and there, it's endemic, right? It's part of what we expect to see. Sporadic, occasional. This would be like rabies. Very good one. Get bit by a dog and get rabies. It's just occasional cases here or there, usually for a weird situation. An epidemic is anything deviating from what's expected. Okay. So anytime that we have, like if we had a whole ton of cold, everybody getting cold, everybody has cold, everybody coming in with cold, everybody constantly having a cold, and that being a thing, that would be an epidemic then. It may have been endemic at first, but if it starts getting out of what we expect, that's an epidemic. If we have a new thing introduced, then we haven't usually seen it before, like when COVID came. In that area, in that time, it would be an epidemic. Right? We expect to see zero of it at first, and then it became a problem. So epidemic. Pandemic is across continents. If you cross a continent, it's pandemic now. All right, I'm not really going to go over this. It really has to do, I'm not going to talk about that one. History of diseases, susceptible hosts, people who can be infected, people who have disease. This more or less just associates with the disease progress that we saw in chapter 13. So not as interested. We get into screening and testing and stuff for these guys. You're gonna learn more about this whenever we get into chapter, what is it? Whatever it is, 18, and diagnostic tests. But we are gonna mention accuracy of these guys. So screening tests, usually screening tests are going to be done to say, it looks like you might have a problem here. And then you do more definitive testing after that. And that makes sense. Pap smear, good one. Let's see if you have any cells that look like they could potentially be cancerous. Oh, looks like these are all normal. We don't need to investigate further. Or, oh, these ones look a little bit abnormal. Maybe we should do a colposcopy or do a biopsy or something like that to see if there's actual cancer progressing here. So screening is just screening, and you'll have to go in depth into more specific tests later. These terms, specific and sensitivity. Everybody hates it, but you have to know it. Okay. I like to start with the sensitivity one because I feel like if you're really sensitive, you will be able to identify any positives anywhere. So maybe this person's like obviously really sick and whatever, and I can say, okay, this person's sick. But the person back there just got a little bit of a tickle in their throat. I should be able to say that person is sick too. So sensitivity deals with making sure you are getting all of the possible positives that there could be. Identifying that somebody has COVID, even though you only picked up one virus in their sample. Okay, so that's sensitivity. Specificity is dealing with the fact that you want to be sure the things you're calling positive are only COVID. You're not accidentally saying, oh, this person's positive, but they only, so that's what that's talking about. So you want both going on. These are not against one another. You want both of these as much as you can. You want to be as specific as you can, only targeting the thing you're looking at. You don't want to call anything else positive, but you want to be sure you're calling all positive too at the same time. Right? So that's the idea with these terms. And then let's just explain them in words relating to whether you're avoiding, avoiding or recognizing false positives or negatives. I don't know. what you find more helpful to think about it, but that's the idea. Okay, we already talked about Koch's postulates and cause. A lot of times now we use like PCR to look at that. Correlation is not the same thing as cause, just because, so you have to be able to rule that out, right? It's just like with murder. It's just like with murder, right? Just because you walk into a room and I'm standing with blood on my hands and like, you know, Dr. Tim is there dead on the ground all bloody, does that mean that I killed him? No, right? Or maybe, yes, maybe, but it doesn't prove it, right? So you have to go into the depths of it and be sure that you're saying that you're accusing this person, or was she just down there trying to help him and revive him? You don't know, right? So it's that same concept. Correlation versus cause are not always the same thing, but they can be. So a lot of epidemiological work is trying to parse out those two things or see if they are working together. So we do studies, and usually it's going to be randomized control trials. So picking out random populations of people and looking at specific aspects of those groups. Cross-sectional studies. Here we're looking at maybe how people were exposed, like they were at this one movie theater. And they got, I don't even know, zombie virus. Let's just say, I don't know. I don't know. So those two factors, I'm going to collect those from Sunday at this movie and collect the data from that and see, okay, these people were exposed in this theater and these people had the virus at this time in this location. But it doesn't look at what occurred first necessarily. So these all have uses. They don't tell you the same information. Depends on what you're looking at as far as what question you're asking, okay? So looking at a cross-section of what's going on with that illness, looking at the fact that they were, you know, how they were exposed and maybe what disease you're talking about. It doesn't always have to be those two factors, but it's just an example that it's giving. All right, case control studies. Group of people categorized if they have a condition or not. Have or not. And then they're both asked about certain exposures to see if one exposures stands out. So that's like taking somebody being like, oh, these people have a lot of what looks like zombie virus in this classroom. Okay, well, let's take this whole classroom. There's people that have it and those that don't. And then, okay, did you drink from this fountain? Did you eat this sandwich? Did you blah, 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 and try to see which one lines up with the people that have it. They were exposed this way versus people that don't have it. They didn't have that same exposure. That's what that's talking about, okay. Breaking it down. And then a cohort study. People without the condition are enrolled and studied over time. We're going to study the negatives and follow any exposures to see if they become positive. So if you have people who are negative and they enroll into the study, seeing like if they all go drink from that water fountain, the ones that drank from that water fountain that were negative became positive. The ones that drank from the other one that were negative never became positive, for example. Okay, so watch them over time to see if any exposure stands out that way and causes them to get sick. They don't advise them to do any specific thing. They just follow what they're doing and see if that answer comes to them. All right. What was left of this? I don't even know. Surveillance, you guys know what that would be. Just trying to collect data. There are some diseases that are required by law that you have to report. There are certain kinds of infectious diseases associated with healthcare, like catheter associated or surgical site and things like this that they will want to track within the hospital to maintain cleanliness and make sure they're not going too far out of what's expected and appropriate for that hospital situation. So they have universal precautions in place for any given situation to prevent spread of these things. There's a difference between medical asepsis and surgical, as far as medical being like washing your hands and things. Surgical, you want to get as sterile as you possibly can. You don't always need to be as sterile as possible, right? It would be ridiculous to do that all the time. So maintaining cleanliness versus being as sterile as possible. That I feel like it's obvious. I promise that we're almost done. Are not, this is how you say it, are not, okay? Are or are not, are not. This is the reproduction rate, okay? So what does this mean? That means that if Sally, so if one person is going to infect 5 people while sick with COVID, that's infection. Anyways, how many people are you likely to infect when you're sick with the thing? That's what R naught is. That's all it is, okay? You got the flu and you're about living your life, the average person's life. How many people are you likely to infect during the course of your infection? And you can control that number if you are, social distancing and things like that. But if you're not, this is usually assuming there's no protective thing going on, no, what's I'm talking about, vaccines or anything. Okay, case fatality rate, this makes sense, yes. People who are gonna die, of all the people that are infected, how many of them are gonna die? Case fatality rate. This is just showing the comparison between some deadliness, which is the case fatality rate, versus the communicability, which is R naught. Most communicable, look at these guys up here. We have measles, man. Measles is like one of the most communicable diseases ever. But you can imagine that rabies is not going to be very communicable. It's going to be needing to be bitten. So it's down there on communicability. But the deadliness of rabies is up there with HIV, for example, right? So you can look at these and compare these. Anyways, vaccines are there to provide herd immunity. Herd immunity is whenever we have enough people vaccinated to keep the rest of the population safe. So if you are talking about measles, it's so communicable that you'd want to have a ton of people vaccinated because it's more likely that if somebody comes along that is sick, that they are going to get it. Less communicable diseases, you don't need to have as many vaccinated to protect the population. So that's kind of what this is referring to, but that's why herd immunity is a big deal. Having herd immunity means if we have everybody vaccinated, you're less likely to get people who can't be vaccinated or who aren't vaccinated for whatever reason, and get them sick. But the more people don't get vaccinated, the more likely we're going to keep spreading it and maintaining it in the population. So herd immunity. This is the process for approval of new vaccines. You guys are, I'm not going to test you guys necessarily over it, but there's some information about it if you're interested, about the different phases involved in those. And then it's talking about just the different steps involved in that. Vaccine injury, you can report these to the government if you're interested. And if you have any side effects from any vaccines, they'll keep track of that because they do want to know about that. Emerging diseases versus re-emerging, brand new diseases versus stuff that we're seeing again because of global warming, for example. We know these. Neglected parasitic infections. These are parasite infections that we see in other countries that aren't getting enough healthcare attention. And then the ones that are here in the United States. These are the ones here in the United States. Chagas disease, neurocystic sarcosis. This is tapeworm that you eat the actual little tapeworm guys that can give you worms in your brain, basically cysts in your brain. Very serious. Toxocariasis, I don't really know a lot about it, but can lead to blindness to worms. Toxoplasmosis, this is don't touch your cat litter if you're pregnant, can lead to miscarry and stillbirth. So like when people say it, don't do it, like it's not a joke. It's not a big deal at all for people who are healthy otherwise, and the mother would never get even sick, but it can kill babies. Yes.
My mom had to quit a vet clinic partially because of the fact that she was pregnant. But
Yeah.
also her boss just sucked. But
You'll for sure see with that, you'll for sure see, this is all over the place. Toxoplasma is at like a lot of places, like probably your cat has it, almost certainly if you have a cat. So they maintain in the cat population. And then trichomoniasis, sexually transmitted disease, protozoan. So anyways, that's it. Oh, sorry, I just didn't want to like leave out that stuff and like be like, you guys watch about that because I hate being that guy.